Okay, hey everybody. I think we're going to go ahead and get started. So welcome to our first webinar in our summer series. I'm Dr. Dennis Mischler. You can call me Dr. Mischler or Dennis if you really want. This is new for me, and it might be new for you guys. So if somebody could, or maybe a couple people say hi in the Zoom chat, just type in there and maybe I can see your messages. Oh, great. Thank you so much. Awesome, guys. Thank you. Nice. All right, great. Thank you, guys. So if you have any pressing questions, I'm going to need you to type in the chat right now because I can't see if you guys raise your hands. So feel free to do that. Hello, everybody. Yep, great. Cool. So let's go ahead and get started. So yeah, so iGEM summer webinars. So I've titled this Molecular Biology, Measurement, and Modeling. And for this first one, we're going to have a pretty short webinar today, just introducing what we're trying to do this summer and telling you guys a little bit about it, and then hopefully taking questions that you guys might have. And, oh, yes, hello, Rochester, New York. All right. So let's see here. Let's go ahead and get moving. Boom. Okay, so this summer, the measurement committee decided that we are going to have this webinar series to hopefully help everybody out and to engage with the rest of the community. Because we're all locked at home. I'm in my bedroom here. We're all at home, lonely. We got to interact somehow. So this is the measurement committee from 2019. We're about twice as large now. And we've got members from at least North and South America, Europe, and Asia. And if you're interested in the future and being part of the measurement committee, let us know. We're always looking for more people to join and give us some ideas. We've got faculty, grad students, research scientists, iGEM alumni who are no longer participating as undergraduates. Got a nice diverse group of people. So about me, there's me. I have my mouth open. My hair was a lot shorter back then. My hair is long now. Haven't had a haircut in many months. I'm a professor at University of Texas in Austin. So that's here in the USA. And my emphasis at the university is really research and education with undergraduates. So year-round, I work with undergraduates. We work in a research lab. We learn how to do research, education in that area. Okay. I've been an advisor and a PI for the UT Austin iGEM team since 2014, and I've been a judge at iGEM since 2015. All right. And like I said earlier, I kind of need a haircut. I noticed this the other day. I was like, oh man, my hair's so long. So building an iGEM project is really tough. Right? Science is tough. I know this. You guys know this. It takes a lot of work. And we have normally tried to be there for helping students not just do their projects, but to collect good data. Right? So the measurement committee, our goal is to get good measurement data that can be analyzed and used for making conclusions. Right? And so to get to the point where you can collect good measurement data, you first have to have a project, and you first have to be successful in creating the project. So making a DNA construct, putting the DNA sequence into your organism, and then having your organism do some functionality that you can then quantify, whether it's fluorescence or just growing or doing something else. And during this process in science, we always talk to each other, right? If I'm in the lab, I'm always talking to my neighbors, the lab's next to me, to my students, to other professors, getting their input. And for iGEM with undergraduates, I know a lot of you guys are doing science for the first time. This is the first big research project you're working on. You've come up with some cool idea of your own. And we want to make sure that we're there to help you guys out if you need us, okay? So if it's a concept, a technique, you just want some outside feedback, we're hoping to be a resource for you guys this summer. We want to be there to help you design and plan out your project. And potentially even talk to you about some of the experiments you're conducting during your iGEM journey. And hopefully by helping you guys out earlier in the process, we can help you think about what types of controls you might want to run, which techniques would be better for your experiments, things along that line, okay? We want to make sure you guys are successful, and we want to make sure that you guys could collect robust data, right? If you happen to be able to be in a lab this summer, great, let's help you guys be successful. If you're not going to be able to get into the lab this summer, maybe you're going to get into the lab next year. And so we want you guys to plan your experiments now so that you can be more successful when you get to the lab in the future. Okay. So this year's journey is going to be really different. Normally, every year teams get into the lab, they get some experiments going. Things might not have worked, but maybe they did. Looks good. You know, it's a good experience, whether you're successful or not. This year, though, a lot of our labs, including mine, aren't open. No one's going to be able to get into the lab to do any experiments until at the very earliest fall, or maybe not even until 2021. Okay. And so we have to recalibrate how we as a measurement committee can help you guys out. And what we decided is that we could potentially help you guys out by creating a series of webinars over the summer. And these webinars are going to focus on molecular biology, how to do modeling, and how to do measurement data, how to do measurement collection. So basically, how do you collect data in a way that you can actually then use the data robustly for making an analysis, okay? So even though many of us are stuck at home, we're still hoping to be an active community, interacting with you guys digitally through webinars or other formats. And we're hoping that you guys are interested in interacting with us, okay? This is a photo of the iGEM Jamboree last year in Boston, 2019. This is the iGEM from above photo that you may have seen before. Every year we take it, every year it gets a little bit bigger. And so, you know, I want us to all realize we're still a community, even if we don't see each other in person this year. So our webinar format is going to happen over the next couple of months. And the webinar format is going to have 1 or 2 webinars each week. And the first one that we're going to have, the first real webinar is next week, Tuesday, June 16th, 10 a.m. Boston time, so this exact time. And this first webinar is going to be titled Getting Started. And each webinar will have, you know, we're not going to really try to do one straight webinar like with like nonstop talking. We're going to try to break it up into chunks. They're going to each have hopefully 3 15-minute chunks. And so for next week, for Getting Started, Alexi's going to give the first block of time. And it's going to be, how do you plan and design your project? So where do you get started? And so he's going to present that information for about 15 minutes. And at the end of that first 15-minute chunk, he's going to allow for questions. He's going to take questions from the audience, give some feedback. You guys are going to do that for a little bit. Take a short break, and then we'll transition to the second block of time. So Ian will then lead a little bit of a webinar on databases. So how do you use databases to get maybe DNA sequences, or to decide what sequences or enzymes you want to work on? And then after that time is done, we'll have some questions. And then Sonia will lead the last section. So talking about cloning strategies, how do you choose a cloning strategy that's better for you? Okay. And so each of those chunks of time will be about 15 minutes with questions following. And so overall, it'll be about an hour, maybe a little bit longer. Each week we'll have a different topic, and hopefully these 3 blocks of times within each webinar will make a lot of sense and present a coherent story. But we realize not everybody needs every little bit of detail within a single webinar session. You might think, hey, I know how to use databases really well. I do that a lot. I've done a lot of that stuff. But maybe you really just need that cloning strategy. You haven't really thought about alternative cloning strategies. And so what we're going to do is, after the webinar is done, we're going to put the webinar content online in these smaller blocks. So you'll see not just one video from that webinar for week 1, you'll see 3. And each one will have that different focus. And so that way, if you ever want to go back and find that precise information that was of interest to you, you can go find that, right? So these shorter segments are going to be uploaded. They'll be available to everybody after the webinar. And the webinars are going to focus on concepts, techniques, and experimental design. During our webinars, though, we're also going to link to other online content. We're not going to try to go into every little detail about each technique or concept we talk about. Well, but we will reference you or refer you to other videos or content that you might find online that could help you go further if you wanted to, right? We're also going to have wiki pages related to each webinar that will contain a lot of the same information there with the links in our videos, of course. Finally, we're going to also have office hours. The first office hours are actually today. And so during these office hours, which will happen every other week, you can come in and talk to me, talk to another faculty member, a graduate student, somebody who's been doing synthetic biology for a while, and ask them some questions about either the webinar topics or about something that's important to you that's related to genetic engineering and or measurement, okay? So here's our webinar series schedule. So this schedule is going to get online, I think, in the next day or so, but this is our schedule. So week 0, that's this week. We're doing this one right now. And then you can see here we have 8 weeks of webinars. Notice there's 3 colors. There's an orange pairing, those 2 webinars will be focused on modeling. So basically introduction to doing some modeling and then a little bit more advanced modeling. And that'll be during week 2 and 3. And you can see those times there. Notice that the modeling talks in orange are going to be at 7 a.m. Boston time. Now that's kind of early. For me, that means it's like really early in the morning. I'm not going to be awake and watch that webinar, but I can find it online. After the webinar is done, it'll be up there as a video and I can go back to that video and see everything. Okay. We're also going to have in green a pair of webinars on fluorescence. So looking at fluorescence with plate readers and then looking at fluorescence with flow cytometry and some of the considerations you should think about before you do those experiments. Okay, and that'll be in July. And again, those 2 will be at a little bit of an earlier time, 7 a.m. Boston time. The rest of our 8 webinars, though, will all be related to molecular biology. So getting started is the first one, and then we'll get into DNA parts and basic molecular biology, go into assembly techniques in week 3 and 4. And then in week 5, we'll talk about cell-free systems. So maybe you don't want to even use cells. Maybe you just want to use a cell-free system. Those are really up-and-coming platforms. And then week 6, 7, and 8, we'll talk about some other topics, transforming your DNA into cells, whether it's E. coli or some alternative system. And then also sequencing your constructs, which is really important. That's part of those, that's one of those non-quantifiable measurements, right? You need to really make sure you have the right DNA sequence. You need to make sure you have the right construct. And we'll talk about CRISPR and protein characterization towards the end of the summer. Okay. So these are the topics. And again, all of these webinars will be recorded and they'll all be put online so that you can find that material after the fact. Okay. So that's our series. Now I want to talk real quickly about molecular biology, because molecular biology is, you think you have to be in the wet lab, but you don't, not always. A lot of molecular biology starts using a computer, using a web-based application or some sort of tool that allows you to design a DNA sequence. So if you need to order primers for PCR, probably going to have to use a web app of some sort. If you want to plan out your Golden Gate assembly or BioBrick assemblies, probably going to use a web app first to make sure all the sequences match. If you want to do an alignment for your DNA sequences, you've got to use some sort of alignment tool, which is going to be, again, a software of some sort. So during the summer, we're going to be using various web applications to help you guys learn about these processes. At this point, I would get a little worried if I were you. I'd be like, oh my God, there's so many different ones. What if they're not using the tool I use? So we've thought about that. And what we decided is we're going to try to focus on 4 of the main ones: SnapGene, Benchling, Genius, and hopefully SBOL. And what we're going to do is during the summer, we're going to have at least one of our presenters, so me and then Renee and Ian also, we're going to make sure that every week, every time we introduce a new computational web app tool, we make sure we use our program with that technique. So for designing primers, we're going to make sure that we show you a demonstration of designing primers with each of these different programs. For Golden Gate assembly, same thing. For BioBrick assembly, same thing. Gibson assembly. All these different DNA assembly techniques where you would use your web app to design the DNA sequences, we're going to make sure we have an example of doing that with each of these systems. So that way you can just choose the one that you prefer at the very beginning and just go with that the whole summer through. If you choose Benchling, you're going to end up seeing me do a lot of demonstration videos for you. But if you choose SnapGene, you're going to get Ian, and if you choose Genius, you're going to get Renee, and we're trying to get somebody to do SBOL. Okay. And so the idea here is you don't have to learn all these different programs. You can just choose one that you like a lot and stick with that the whole summer through. Okay. All right. So what's next? Well, next week, getting started. And you can see here the overview of that. This is the next webinar. Alexi, Ian, and Sonia will be leading that topic next week. And we're going to start with basically just saying, you have an idea, let's get a DNA sequence, and what do we do with that DNA sequence? Okay. That'll be same time next week. That one will be a longer webinar. It won't be this short little block today. It'll be 3 blocks back to back. Okay. You can always contact us at our email here, measurement@igem.org. We have our office hours. These are going to be today at 3 a.m., so that one already happened, and then 1 p.m. Boston time. And these will repeat every other week. Okay. And if it turns out that's not enough, if you guys want us to do stuff more frequently, let us know and we'll try to make that happen. Okay, but for right now, that's our starting point. All of our webinar material can be found online at the iGEM website if you use measurement as the hub. And our webinars will also be able to be found at the calendar event. So you can go there to see when they're going to be scheduled. Okay. So all that material is going to be up there in the next day or so. If it's not up there right now, give it a little bit of time. It should be up there within the day. Okay. And I think with that, that is everything I wanted to cover for today, guys. So what I want to do now is just sort of open the floor for people to ask questions and yeah, let me know if you have any questions or anything like that. And I will try to do my best to answer your questions. Oh, there's a Q&A, sorry. Okay, so we have a question. Sorry, guys, this is my first webinar, so I was hoping to be able to actually call on people, but it looks like I can only see typed answers. So I have a question here from Michael. He's from Ghana, and he asked about where can we access the videos. So we're going to get those videos up there in just a little bit, and then we'll let people know where it's going to be accessible. So it might be uploaded to YouTube, which would be really easy, but it might also be uploaded to an additional site. Yep, no problem, Michael. Will the presentation, yeah. So the presentation slides will, this is from, I'm not sure how to pronounce the first name exactly. I'm gonna say it's Coke, maybe, G. Lee. And I'm not sure where you're from, but if you let me know where you're from, I'll say where you're from. The presentation slides will be available. We're gonna have a PDF of the slides, and those will be uploaded to the wiki as well. So if there's links in there that have important information, you can click on the PDF link and that'll get you to where you want to go. Okay. Other questions, folks? And there's a Q&A thing. I don't know if you're trying to ask questions, you can either use the chat or you can use the Q&A part to do that. This is from Lin Hong. And again, I apologize if I mispronounce people's names. I will try my best to always get it correct, though. You can't, yeah, absolutely. You can totally contact the, she's at, so Lynn is asking if they can contact the committee to ask for resources. The answer is absolutely yes. And you have a question here about modeling protein, DNA, RNA. And yeah, that would be absolutely something you can ask us about. We'll be happy to give you our input. Jake and Alejandro are the two members of the measurement committee who will be leading the modeling webinars in a couple weeks, but you could ask them questions today at office hours or through email. That would be totally cool, right? And in the future, guys, if you have any ability to let me know where you're from in your questions, I'd love to know that just so I know who's watching us. All right, and thank you, Lynn. Next question here is from maybe Uraris. I'm not sure exactly how to pronounce that, but Uraris from Indonesia, or Ayas from Indonesia, is webinar provides certificate. I'm not sure exactly what you mean by certificate. If you follow up with that, I'll be happy to let you know. Other questions? If you're having trouble finding out how to ask questions, just let me know. All right, I don't see any other questions at this time, guys. So if you do have questions, though, feel free to email us at the measurement group, or feel free to pop into our office hours, which will be starting at 1 p.m. Boston time. So that's about 2.5 hours from now. Okay, all right. Otherwise, I'm going to end the webinar. So thank you, everybody, for coming out today. Thank you for showing up to our first introduction one. And like I said, the real content will, got a question here. Real content will start next week with going into starting a project planning, and then from there on to other topics. So question here, this is from Arnaud. He's from Toulouse and in France. and they asked, do you know when the pages for awards website will be updated? You know, I don't know when that will be updated. My guess is it'll be updated sometime this month, though. Remember, one of the big things this year with iGEM that's different is since we don't have a lot of teams that can get into labs, maybe about half of our teams have no chance of getting into labs, the awards and medal criteria are being changed a little bit to be more flexible to allow those teams to participate. So we'll make sure that gets updated. If you have specific questions, you could send them in and we can push them to the right people to get them maybe answered for you. Okay. Thank you guys for asking the questions. I appreciate that so much. Thank you for attending today. And oh, I am out of Zoom or out of focus now. There we go. All right. Thank you guys so much. Have a great day. If you don't pop into office hours today, great. Hopefully we'll see you at the next webinar. Otherwise, have a great summer and take it easy. Stay safe, everybody. Bye.